Hello everyone. I welcome you all to Biotech Made Easy videos. Today I have come up with a video on multiple choice questions on DNA fingerprinting. This is a topic on which invariably many questions are asked in different competitive exams, be it NEET or GATE or any other exam. So I hope you already have a background of DNA fingerprinting, and I have also uploaded one of my videos on DNA fingerprinting, so you can go through that also. I'll give the link of that video also in this at the end of this video. Okay, so let's start the DNA fingerprinting quiz. Let's see what is the first question. The first question is, who discovered DNA fingerprinting? There are four options: Alec Jeffrey, E.M. Southern, Carey Mullis, and Francis Crick. Now you see, all of these are big shots of biotechnology. Out of all these four options, E.M. Southern has given a technique which is known as Southern blotting. Carey Mullis has given a technique which is known as PCR, and Francis Crick is the one, Watson and Crick, who gave the double helical structure, but the answer is Alec Jeffrey. So if you have read this chapter on DNA fingerprinting, you will be able to answer this. So the answer is A. Now let's see what is the second question. Who is father of DNA fingerprinting in India? Now here you see four options are Hargovind Khurana, Ram Rajshekharan, Deepak T. Nair, and Lalji Singh. Now here the answer is Dr. Lalji Singh. Again, in this particular question also, all the four contemporary scientists, means Indian scientists, are given here, so you don't need to get confused. Maybe you already know about the Hargovind Khurana because he got the Nobel Prize, so that one can be eliminated very easily. And then also I would like to tell you that Ram Rajshekharan is a plant biologist, and Deepak T. Nair, he has worked on DNA and RNA polymerases. But the right answer is Dr. Lalji Singh. So here, I have given this question to many of my students at the local exams, not at the national level exam, but in the local exams. And you know what, they get confused. They just don't read in India. And if if one of the option is also Alec Jeffrey, then they always tick Alec Jeffrey. Here there is no confusion because Alec Jeffrey's name is not given here. But generally, if this question is asked, you may have one option Alec Jeffrey. So just you have to write the whole question very carefully. That is my suggestion to you, and not only for this question but for all questions. Then let's see the third question. DNA fingerprinting was discovered in which of the following institutions? So if you know that this technique was developed by Alec Jeffrey and he was working in UK, then it would be easy for you to answer. Otherwise, this is going to be difficult. The answer is University of Leicester, UK. Another question is, Dr. Lalji Singh was director of which institute? Okay, so again, this is a kind of question in which if you know the answer, only then you should tick. Otherwise, there are chances that you may be wrong, because all these institutes, IVRI, CCMB, ITRC, and BPGR, all are the institutes of national repute in India. And the answer is CCMB. Dr. Lalji Singh worked in CCMB, and for a long time he was director of this institute. Fifth question is, which of the probe was devised by Dr. Lalji Singh? BKM, MMK, QMB, CKT. These are the four options, but the right answer is BKM. And I tell you little about this, that Dr. Lalji Singh used to work on snakes, and one of the snakes on which he was working was the banded crate. So this probe was prepared from the satellite DNA of banded crate. So that's how it was named as BKM. M is for minor satellite. Next is satellite DNA is not found in telomere, centromere, heterochromatin, euchromatin. The answer is euchromatin. You might be knowing that the satellite DNA is a part of telomere and centromere, and heterochromatin is something related to telomere and centromere. So these three cannot be the answer. So the right answer is euchromatin, because here mostly the coding sequences are present in euchromatin. Next is, what are the tissues used in DNA fingerprinting? The options are blood, tooth, semen, and all of the above. Now, if these kind of questions are there, mostly the answer is all of the above. Let's see what is the answer. Here also the answer is all of the above. When I was preparing, or I was framing this question, it was in my mind that there are so many biological samples which can be taken for DNA fingerprinting. So I wanted you to know that blood, tooth, semen, all these can be taken. So that's why I have given this fourth option. And according to surveys also, it is said that if one of the options is all of the above, the chances are more than 25%. That means that there are many surveys, and they say in between 30 to 50% chances are that this option can be right. So if you know the answer, very good. But if you do not know the answer and you want to just guess, then you can go for the all of the above answer. But of course, keep this in mind that there should not be negative marking. If there is a negative marking, then you should not take the risk, and even if you are taking the risk, then it should be very calculated. Then next is, how much DNA is identical in all human beings? Okay, now this is again a very tricky question, because you see all the values, they are so close to each other: 99.9, 80.8, 91.5, 75.8, and it would be difficult just to guess the answer, because until you know exactly how much DNA is identical, you cannot find the answer. So let's see what is the answer. 99.9%. And when I was studying and I first time came to know that the human beings have 99.9% DNA which is identical and common, I was really surprised. So if you are guessing, then nobody would guess 99.9%. So it's better in these type of questions that you should not guess and you should not take the risk. Next question is, what are mini satellites? And the choices given are non-coding regions with tandem repeats, coding regions with tandem repeats, non-coding region without tandem repeats, and coding regions found in prokaryotes. So all the answers are a little confusing, a little similar to each other. Let's see what is the answer. The answer is A, non-coding regions with tandem repeats. If you have gone through the DNA fingerprinting, you must be knowing that satellite DNA, whether it is mini satellite or microsatellite, it's always non-coding. So if you know that, it would be easier for you to eliminate, because here you can eliminate coding regions B and D. So because in B and D there is written that it is coding region, so that can be eliminated. Now satellites, they have always tandem repeats, so this you should remember, and that's why the answer is non-coding regions with tandem repeats. The other one was without tandem repeats, so that's not possible. What is the full form of VNTR? VNTRs are actually mini satellite, and these are the mini satellite DNA on which the Alec Jeffreys technique was based on. The answers are various non-coding tandem repeats, variable non-coding tail repeats, variable number of tandem repeats, and various number of tail repeats. If you see the answer, it is C, variable number of tandem repeats. In the earlier question also we said this is non-coding region which has the tandem repeats. So here you see it is variable number of tandem repeats. That is the answer, correct answer. Question number 11 is microsatellites are also known as, choices given are VNTR, STR, RFLP, RAPT. You know, these are all molecular markers. All of them can be taken for the DNA fingerprinting. But as regarding the microsatellite, they are known as STR, that is short tandem repeats. So the correct answer is STR, and you know, in these type of questions, just don't guess. You know, you should know the answer. Okay, question number 12. The repeats in microsatellite are about 50 to 80 base pair, 10 to 60 base pair, 2 to 6 base pair, or 1 to 12 base pair. So these are 4 options, A, B, C, D. What is the right one? Right answer is 2 to 6 base pair. You know, as the name indicates, microsatellite, these are very small repeats. So here out of these 4, the right answer is 2 to 6 base pair. But to answer these kind of questions, you must know what is the number of repeats. You know, sometimes in some of the books you may see that 2 to 8 base pair is also written, but out of these 4, 2 to 6 is right answer. Which of the following is part of DNA fingerprinting process? Now the options are chromosome walking, western blotting, southern blotting, and thin layer chromatography. If you know the mechanism of the DNA fingerprinting, it is a very easy question to answer. But if you do not know the mechanism, then it would be difficult. Answer is southern blotting, because in this whole process you do the electrophoresis, and after electrophoresis the DNA bands are transferred to the nitrocellulose paper, which is more robust, and that is southern blotting, which is very important part of this mechanism. So C is the right answer. Next question is, for DNA fingerprinting, which of the following step is not required? And see what are the choices. Collection of biological sample. I think you all must be knowing that collection of biological sample is, you know, must. You can't do without sample. Electrophoresis. Electrophoresis is also very important part of this mechanism. Isolation of DNA. You have to do isolation of DNA also from the sample, because its name is DNA fingerprinting. So you have to do this whole process, DNA fingerprinting, from DNA. So you need DNA. And then the last option is given here is cloning of DNA. Cloning of DNA is not needed here. In some cases, the PCR is done to amplify the DNA, but not cloning of DNA. So the right answer is cloning of DNA. Other 3 are must. These are the important steps of DNA fingerprinting. Next question is, DNA fingerprinting is mostly used in which of the following? Here the options given to you are forensic sciences, reverse genetics, chromosome mapping, population genetics. And this is an easy question. You all must be knowing that the answer is forensic sciences. There are different cases of crime, like paternity disputes are there, rape cases are there, then murder cases are there. In all these cases, the DNA fingerprinting is done to identify the culprit. This is, here I would like to say this is one of those biotechnological techniques which you can hear about in newspapers and and in different channels whenever there is a high profile case. So in all these days it has become a routine to take the help of DNA fingerprinting in solving the forensic science cases. Next question is, in DNA fingerprinting, which of the following is labeled? The answers may be one of these four. DNA sample? No, DNA sample can't be labeled, linker molecules, here we do not use linker molecule. Then other two left are probe and electrophoretic gel. The answer is probe. So when Alec Jeffrey was doing this experiment, he labeled the probe. In some of the cases these days, even primers are also labeled, but primer is not given as the option here. So here the answer is probe. Now come to the next question, question number 17. Which of the cell type cannot be used for DNA fingerprinting? Okay, now this is a little tricky question and a difficult one also. The options given are parenchyma cell, erythrocyte, white blood cell, and liver cell. You may think that blood is taken as a one of the biological sample, in fact very commonly collected sample. Then and erythrocytes and white blood cells both are present in the blood, so these you will not eliminate, but actually the right answer is erythrocyte. And erythrocyte, you must be knowing that this is red blood cells, but here you must note that in red blood cells or in erythrocytes no nucleus is present, and if no nucleus is present, that means no DNA is present, and for DNA fingerprinting you need the DNA sample. So that is the reason that erythrocyte cannot be taken. All other blood cells, white blood cells, whatever else is there in the blood, all those components can be taken. And liver cell, parenchyma, they all have nucleus, so no problem, they all can be taken, but not erythrocytes. Next one is, DNA fingerprinting is not successful in which of the following? The answers given here are, if individuals are identical twins, if the sample is contaminated, if there are more than 5 suspects, and both A and B. So you have to choose one. Let's see what is the answer. The answer is both A and B. Why? Because actually when the identical twins are there, because they are identical twins are monozygotic, one zygote is formed, one egg and sperm are fused, and from that zygote that same zygote is divided into 2 individuals. So, and that is the reason the DNA is exactly same. So it is not successful if the individuals are identical twin, and also if the sample is contaminated. If when you have isolated the DNA and at that time there was due to some carelessness or because of any other reason, if the sample is mixed of 2 individuals, or even the bacterial or fungal contamination is there, any kind of contamination is there, then also the DNA of those organisms, microorganism or the individual, that will give the misleading results. So naturally the answer is both A and B. And as regarding the C, if there are more than 5 suspects, doesn't really matter. Even if there are 10 suspects, more than 10 suspects, 50 suspects, no problem. You can do the DNA fingerprinting, can make the band pattern and you can compare. So that's not the problem. Come to the next question. On the basis of comparison of band pattern from crime scene, who amongst the following is the culprit? Okay, so here we have given you, 4 band patterns. See, the first one is the one which has been taken from the crime scene, and the other 3 band patterns are from suspect number 1, suspect number 2, suspect number 3. So what we have to find out? We have to find out the culprit. That means out of these 3 suspects, one is going to be present at the crime scene. So what does that mean? That means out of these 3 suspects, one's band pattern will be exactly same as the band pattern of the DNA which we have found from crime scene. So you have to now compare, and when you will compare the band patterns, you see different bands are present at different locus. So here you can see the answer is suspect 2. To make it more clear, I have shown it by green color. So now you can compare this band pattern along with this band pattern which you got from the crime scene, and you see all the bands are present at the same position. They are exactly matching. See, they are exactly matching. So that means this suspect 2 is the culprit, and the chances are that this culprit was present actually at the crime scene, and that's why the band patterns are exactly matching. So that's how you can do the comparison. Even if there are more suspects, then also the comparison can be done. And yeah, one more thing I would like to tell here, that here one of the options is none of the above, and generally it has been observed that no one gives this none of the above answer. Means like most of the time this is not the right answer. When I was framing this question, I wanted you to find out who is the culprit. I wanted you to do the comparison and find out the right answer. I never wanted to have the answer as none of the above. And surveys also say that most of the cases this none of the above should not be guessed. You should not take the risk of guessing none of the above as the answer. Okay, so now the last question of the quiz, and that is, which individual is father of the child on the basis of the band pattern shown below. So here the five different type of band patterns are shown, and what you have to find out? You have to find out the father of the child. So first you see the band pattern of the child. Here if you see, the bands are shown in 2 colors. One is red and one is blue. Red colored bands are similar to the mother. The first one is the band pattern of the mother of the child. So as you know that any child gets half of the DNA from father and half of the DNA from mother. So you know what happens? The half of the bands are similar to mother and half of the bands are similar to father. So that is what happening here. Out of these 6 bands, 3 bands which I have shown from red, they are matching with mother. So that means these 3 bands you just eliminate. These 3 blue colored bands are left, and these 3 bands you have to match with these individuals to find out which individual is the father. So let's go for the answer. Here you can see the third individual. In the third individual, 3 bands which I have shown by blue color, they are exactly at the same position as in child. So that means these 3 bands are matching with the child. So that means this person is the father of the child. So here you can see the answer is 3. So that's it about the quiz on DNA fingerprinting. I hope your chapter on DNA fingerprinting has also been revised, and I'm sure that these questions are going to be beneficial for you in any of the exams which you may go for. So I wish you best of luck and a bright future.